Okay, everybody, we've almost made it through the end of the first week. Everybody ready for a first weekend? Just about, I know I am. All right, so today we're going to move on a little bit from that sort of general overview, introduction to ecology that we talked about on Wednesday. And today, it's probably going to run a little bit beyond. We'll probably still be talking a bit about this on Monday because this is probably one of my favorite lectures in this course, in this series. I still remember several years ago when I was first taught this same material as an undergraduate. It was one of the things that sort of really piqued my interest in ecology and dragged me into a career studying ecology and biology. And what we're going to discuss is ecology and evolution, how the ecological side of evolution. And we're going to do this, it's nice, it's a general overview. We're going to talk a bit about the ecological mechanisms. But right the way through, as I'm talking and as you're trying to interpret the information I'm giving you, remember what we talked about on Wednesday about the scientific method. And think about this as the mechanism through which we can get to all the deductions, all the conclusions that we reach on the various different examples that we talk about. Because what we're really going to touch on here is the mechanisms through which evolution happens, particularly looking at natural selection. We're going to talk quite a bit about selection, ecology, the role of ecology or species ecology or populations or individuals ecology in determining the natural selection, determining which individuals in a population do best, which individuals in a population are more likely to go on to reproduce. We'll look at how this works along particular gradients. If you're a plant or a fish living in one area, how does that change if you move to a slightly different area, or if you're a plant in a slightly different area? Later on in the course, we're going to talk a lot about climate, habitat, environment, things which determine what type of plants or what type of animals will succeed in a certain area. But today we're really looking at the mechanisms through which that can have an effect on what species are found in an area, and also what type of individuals within a population are found in an area. And as we'll see, that really is the core driver of evolution. We'll also touch a bit on ecological speciation. At what point does evolution take us to an extent that we can say or we can expect that one true species, that what we've moved beyond is just variation within a population or different variants of a population to two or more actually distinct species. And then this will probably take us into next week. We're going to look at eco-evolutionary feedback and how essentially what I'm going to talk about the majority of times of this lecture anyway is how ecology drives evolution, but also evolution can drive ecology. And we'll see that cyclical effect as we move on a little bit further. So, to start at the start, nothing in biology makes sense, or biology only makes sense in light of evolution. And we know that adaptation by natural selection is the main driver, or at least the main ecological driver, we'll see in a moment, of evolution. And Darwin, so this is one of the first issue of Darwin's book on the origin of species by natural selection. And this short text here is, if we were to summarize the main conclusions from Darwin's work, from the work on natural selection, the earliest work on natural selection, we could do it, and this is the piece of text here. If traits of organisms have heritable variation, and that variation affects survival or reproduction, then trait values will change across generations. So there's three key pieces of text within that, three key statements within that. First of all, we need organisms must have heritable variation. So if you have a different hair color or skin color or body shape or mouth morphology, as we'll see, that must be heritable. It must be something that if you have this trait, your offspring will also have this trait. If that variation in that trait affects survival or reproduction, we'll see in a little bit later on, we'll look at some examples of birds and fishes and their mouth morphology. Essentially is what allows them or determines what they can eat. So it's a really key trait, and it will affect if you have a mouth that's one particular size or a particular shape. It will determine the type and amount of food you can eat. And that is a key aspect determining whether you're going to survive and be able to reproduce. Provided you have these two things, provided you have heritable variation and variation that affects survival, then you'll get a variation in the frequency of specific traits across multiple generations. We'll talk a little bit more exactly about how this works. But essentially natural selection, this is a very simple mechanism. It's simple, it's powerful, and it's inevitable once we have this a system that's aligned in this way. In your introductory biology courses last year, you would have done, everybody's done some introductory genetics? Yep. You know that mutations, variants in genes, that comes from genetics. The selection part of this process, what happens once we have this variability, that's driven by ecology. We can take this sort of walk-through path to see how a population with this light grey trait could turn into a population dominated by this dark grey trait, sort of hypothetical walk through evolution. This individual, this population has offspring with a variety of traits. A grey dot can give rise to a white dot, a grey dot, or a darker dot. We can think of this could be body size or mouth morphology, any trait. There's a selection gradient against white dots. Although we've got this variability through mutations, the only ones which can reproduce, which succeed to reproduce, are the darker dots. And as we go through multiple generations, there's consistently selection against the lighter dots. The darker dots ultimately come to dominate the population. The genetic side of things is what gives us this variability. We're not going to talk any more about that in this course. What we're going to talk a lot more about is the ecology, the selection side. What traits lose out, which traits succeed, and why that happens, and how that happens, and what occurs when that happens. I'm going to talk, one of the main examples I'm going to work with for certainly the first half of this lecture, we're going to talk a lot about birds. And these are an example of species of finches, commonly known as Darwin's finches on the Galapagos Islands. And these are one of the key models for natural selection. They're one of the regions which really were fundamental to Darwin's work when he was formulating his hypotheses based on his observations. And there's been an area rich in evolutionary research since then. And the first thing to note about these, this is one species of finch. And we've got a plot here, this is taken from your textbooks, of beak depth. So if you gather population size, we don't. If you gather around about 1,000 finches, take a big sample of finches from an island in the Galapagos, measure their beak depth from the top of the beak to the bottom of the beak. How big is it? They won't. Every single bird you measure won't have the exact same size beak. They'll all be kind of close, but if you do quite a few, you should get that kind of a distribution. So here, the number of finches that have a beak depth, the mean value is around about 9.5, but there's quite a few of them have 10, there's quite a few of them have 9, quite a few of them have 11, fewer have 8, some have 7, 1, 2, maybe have 6. So we've got a range in values. We call this, we're going to talk a lot more about this in subsequent lectures on statistics. We call this like our normal distribution. And any trait you look at will have this range of values. The other thing that's crucial here is what we see in this plot here. That parent beak depth is a really good predictor of offspring beak depth. Or beak depth, sorry. So, if you have a parent, if you have two parents who have a small sized beak, the offspring has a really good probability of having a small sized beak. Whereas if you have two parents whose beak depth is 11 millimeters, their offspring's beak depth is 10 or 11 millimeters. Two different lines here are two different years that this analysis was done. And we see that this trend is robust across years. So this is really good data, really strong data with which we can test hypotheses about ecology and evolution. Okay, so we've got some of our key factors, our key components of natural selection already in place. We've got variability within a trait. We know that this variability is heritable. And we can guess that this trait is ecologically important. For birds, their ability to feed is determined by their beak shape and their beak morphology. We forget about this sometimes because we can pick up and manipulate foods with our hands and chop them up with knives and forks, and we can get pretty much whatever we want. We can get a whole cow into our mouth because we can use different tools to do it. If you're a bird or a fish, you can't do that. You've got one tool, and that's your beak. And that becomes really, really important. So what happens? We've got a system where traits are variable, this variation is heritable, and these traits are linked to your reproductive success or your ability to survive. This is a system that is primed for a study of evolution. As we go through it, we can study it and interpret it using the scientific method. This is some notes. This is basically sort of what I've already said. So, what happens? This is, let's go forward. Okay, so this environment was primed, ready for a study of evolution. These two scientists who look like a standard granny and granddad, or anything but standard granny and granddad, that's Rosemary and Peter Grant, who are emeritus professors in Princeton, and have been studying this system for around about 30 or 40 years, almost into 50 years, and still working in this area. And they've done a whole host of different types of research and different types of analyses on this. But we're going to talk about one of their main findings. So they started early 1970s visiting these Galapagos Islands, sampling finches, measuring what they eat, measuring their beak size, measuring how many of them there are, understanding this population. And what they saw was that first went there around about early 1970s. Over time, each year they'd go, there was some variability in the mean and range of beak sizes of this species of finch, of this population of finches. What they found was that year on year, beak sizes seemed to be getting smaller. And then in one year, they went there in 1977, and there was a really severe drought. this affected the availability of food. Very few tree species produced seeds. There were fewer seeds on the ground. When they returned, so we're going to hop back here. So this is 1976. This is some of their data that we've talked about already. A nice normal distribution with a mean peak depth of around about 9.5. They returned 2 years later and counted the survivors. After this drought, the majority, so this is, if you look at the sample here, they've got 750 birds sampled in 1976, 90 birds sampled in 1978. Okay? A massive proportion of the population died out due to starvation. There was very little food available for them. Of the remainder, what they noticed was the remainder of the population, the ones that did survive, had a larger beak depth. These were individuals that were able to capture or able to feed on larger seeds and nuts that were from drought-resistant plants. So if we take here and move forward through time, beak depth after 1977, after this drought, jumped up. And as we go forward through many further years, for 5 or 6 more years, the beak depth maintained high, was maintained at a larger size. It's a heritable trait. The population had lost all the individuals with low beak size. Only the individuals with larger beak size were left. Mid-1980s, we moved into an El Nino cycle. A lot more moisture in the air, a lot more, the plants were more productive. There were a lot more seeds available, more plants or more birds. The population started to increase again. Birds with different sized beaks were found due to mutations. And over time, this mean beak size started to come back down again and sort of plateau out. What's happening in here is what we're going to talk about a bit more later on, but it's more this, it gets to this eco-evolutionary feedback. When the bird population started to increase, there were more individuals with smaller beaks, and they were able to feed on the abundance of, there was more food available for smaller sized seeds, larger sized seeds. There was a resource available for the population with smaller beaks, or for individuals with smaller beaks, and the mean value started to drift back down again. So over time, we see this shift in traits, or in a trait, in beak size within these birds, as a response to changes in their environment, as a response to changes within the prey that's available to them. We can bring this back, we can look at this in terms of, here. In one generation, the white dots were selected against, small beak sizes were selected against. Perhaps in a later generation, grey dots were selected against, large beak sizes were selected against. We're seeing in real time natural selection working on the variation within the population. Selection can work in different ways. We can sort of break it down that there's three distinct, say, selection gradients. You can have stabilizing selection. Take, for example, a population here of finches, which when we saw them previously had that nice broad normal distribution. You can think of an example where there may be a limitation on large seeds and also a limitation on small seeds. And the only seeds that are available are medium-sized seeds. So the beneficial or individuals within the population which have a medium-sized beak are able to maximize their resource use, are able to have the highest probability of survival, highest probability of reproduction, highest probability of passing on their gene, passing on that trait, and we see this stabilizing selection, forcing a broad population into a tighter range of disease. We can have directional selection. We're going to look at it a little bit as we move along the cline. We'll move along a gradient. And that will force, again, this normal distribution in one direction or another. Or potentially we can have disruptive selection, where the individuals at either end of the range. So when we looked at the finches, we saw that there were some individuals that had very, very small beaks, 5, 6 millimeters there. Some individuals which had very, very large beaks. We can imagine a hypothetical scenario where the environment may change and a competitive species might arrive that can outcompete the finches for their preferred resource, and the individuals at either end of the range, at either end of that spectrum, are then the only ones that have a better chance of reproducing, a better chance of getting enough resources. And that produces a strong disruptive selection, pushing this normal distribution into two distinct distributions. These are hypothetical examples. We can theorize these. We can put this as a hypothesis, and we can say, okay, well, let's make a prediction based on that. Let's make a prediction based on directional selection. If we're working along a gradient, let's take an example here of a mountain top, or even of a hillside. This represents a cline. It represents a gradient in temperature, in water availability, probably in soil depth, from the base of the foot of the mountain, which is maybe drier, certainly warmer, a deeper soil, higher up the mountain, cooler air, rockier soil. So we've moved on a step. We've sort of we have a hypothesis that we've derived from what we know about natural selection. We've made a prediction here that the traits will vary. There's a selective gradient along here. We can predict that traits associated with this gradient will vary. We can then test that. We can go to the field and measure plants or animals and measure those traits and see how does that support or refute our hypothesis. This is an example from Arabidopsis plants. And these are specifically plants which grow along one of these gradients. So we've got elevation, area above sea level, and above ground biomass. What do we see? Does the biomass of the plant, how does the biomass of the plant change as we go above ground or above sea level? What do you think? We're moving from a warm area with deep soils into a cooler area with poorer soil. Will we get more above ground biomass in this cooler area or less? Less? Let's have a look and see. So, what we see is that at the lower end of the soil, or the lower end of the gradient, we've got plants that have, in this case, it's primarily related to the availability of water, but also that they've got a better quality of soil. They send out more roots to gather more through the soil, to gather more water. As we move further up, there's more water available to them. They can grow more. In this particular plant, we get more growth at a higher range. We could think of a plant with a different set of traits. Okay, for this specific plant, that's the relationship we see. That's our test of our hypothesis. We can take that back and say, okay, well, what about a different type of plant? Well, there will be certainly different types of plants, different species that will see the opposite relationship, that you see no relationship at all. And that allows us to get at the mechanisms behind what type of plants, or what the mechanisms behind how selection is having an ecological effect. We can bring this link and link this cycle back together again. That works along clines. But can we also see, or we also see, natural selection working in a discrete environment, where we don't have a gradual shift in water availability, we don't have a gradual shift in temperature, we don't have a gradual shift in soil or resource availability. We have a very strict demarcation. And that can lead to quite often the evolution or the presence of what we term as ecotypes. And this is an example from some mice, a population of field mice, and they can have a range of hair colors, everything from dark to a much lighter color. And they exist, or they're found. They're found on in a coastal environment, an environment that's made up of sand dunes and fields. Two visually very different environments. We take an overview on this. We've got a sandy environment and a field environment, a light-coloured mouse in the field environment, and a dark-coloured mouse, light-coloured mouse in the sandy environment, dark-coloured mouse in a darker field environment. And the question becomes, or what we want to say is, how will natural selection determine which of these individuals are found, or what type of trait is found in this different environment? Well, if you take individuals from one environment and place them in the other environment, now these are the same population. These can easily interbreed. They can move between these two different habitats. It's not different species, it's just the same species, but slightly with different morphology, with different traits. If we can see these, and we can look at this, and we can see, yeah, I can just about see that one, but that's very clear, that's very clear, it's harder to see the mouse up there. The same is true for predators. If we look at the proportion of attacks in a light environment versus a dark environment, in an experiment, so we set up an experiment, release both types of species, or release both ecotypes, observe the rate at which they're preyed upon. The darker prey, or the darker individuals, are more likely to be preyed upon in the lighter environment. Lighter individuals are more likely to be preyed upon in the darker environment. We've got selection against specific traits in a specific environment. What type of selection gradient is this? Is it stabilizing, directional, or disruptive? Disruptive, yeah. We're pushing, so there's a population here that in a normal environment will have a full range of traits, a full range of hair colors. And into a particular habitat where there's a very high risk of predation for one particular hair color in one region, and a very high risk of predation for another particular type of hair color in another region, we get disruptive selection towards dark hair colored individuals and light hair colored individuals across this habitat gradient. That can take us so far. That allows us to say, okay, so selection can determine what type of plants grow in a certain environment. It can determine what size finches or what beak size finches will have in a certain environment over time. It will determine what colour mice will live on a beach versus what colour mice will live in a field. But at what point can we move this forward and sort of say, can we move to a point where these different ecotypes become different species? At what point can natural selection lead to the diversification of one species into others? And to get to understand this, or to get to this point, we need to think about exactly what we mean by a species. At what point does a distinct or a population that's split into two distinct ecotypes, what point does that become two different species? Typically, the standard definition of a species is a group of individuals of a population who are reproductively isolated. Okay? So if you have a case, say the mice here, they can still reproduce with each other and give rise to individuals with a variety of different hair colors. We've got selection at the phenotypic level, but it's not at the genotypic level here. To get to split from ecotypes, we need to have genetic isolation. We need to reduce gene flow between different populations, between different ecotypes. And this can happen through geographic isolation. So here's an example of salamanders in the Appalachian Mountain Range, where we've got different or a single species which has evolved into multiple different species due to geographic isolation. Different species associated with different areas. They can't move, individuals from here can't pass a barrier, they can't pass a mountain range into that region there. And then molecular evolution will do its work, and these individuals will move in different areas or will develop different traits. But can this happen ecologically as well? Can we get instances where ecological selection is sufficient to drive speciation? And there's some instances, there's still a bit of a hot topic. There's still an argument at what point we can truly get a true species. But some of the strongest evidence comes from some work that we've done in BC looking at sticklebacks. And three-spined sticklebacks are these little fish here. They're a wonderful model species to study evolution. They're small, they're plentiful, they work well in experiments. And they also have some key measurable traits. So in these lakes where they've done some of this work, we can see two distinct habitats. There's a shallow water zone and a deep water zone. And these two habitats have very different, so we can say here, sunlight can reach the lake bed, stimulate plant growth. Here, in the deeper water zone, sunlight doesn't reach the lake bed. You've got two very, very different environments, darker, deeper, murkier water. in these two environments, we've got the same species of stickleback has shifted, has evolved into two distinct ecotypes. A pelagic limnetic form and a benthic form. So the pelagic limnetic form primarily feeds on zooplankton. Okay? Small, tiny little crustaceans. It's quite sleek, slim body, large fins, and if you look at it's in its gill, so most people should know what a fish gill looks like. If you look at a fish gill, on the underside, on the other side of the gill arch, there's all these little hair-like structures, little bony structures. And they're called gill rakers. And they have a really important function. They're similar function to finch's beak size. They essentially capture whatever the fish is feeding on and funnel it back into its throat, down into its stomach. If you are a pelagic feeding fish that wants to feed on very small, minute zooplankton, it's really beneficial if you have lots and lots of long, thin gill rakers. Because you can filter, you can make a much tighter mesh in your filter and capture smaller prey items and feed on. If you're a large benthic feeding fish, you're not feeding on zooplankton. You're primarily feeding in the benthos, in the snails, worms, things like that. You don't need such big gill rakers because your prey items are larger. You don't need that nice fine mesh to essentially when fish feed, they suck in a lot of material. It could be sediment, water, whatever. Use their gill rakers to filter out items at a certain size and pass that back down. What we see in fish in BC lakes and stickleback complexes, that there's a benthic form, also that the benthic form and the limnetic form are adapted to their own environment. Okay, so here we've got growth rate, they've taken individuals in both sides or both forms, put them in a cage, put them in their preferred habitat, others in their own preferred habitat, and measure their growth rate. The individuals in the limnetic form, not very surprising, grow very well in the limnetic zone, grow very poorly in the benthic zone. They don't have access. They're not well adapted to feeding on the benthic prey. The benthic form grow very well in the benthic habitat, but grow very poorly in the limnetic habitat. They can't feed on the zooplankton. They're not capable of getting enough resources to feed on the zooplankton. These two individuals, or these two forms, can feed it, they can reproduce. At the moment, there isn't reproductive isolation between those two forms. They can reproduce and they can form hybrids. If those hybrids are viable, if those hybrids are capable of feeding well, reproducing well, then an intermediate form between these two would be viable in the lake. This intermediate form would be able to compete well with benthic and limnetic individuals. And this complex, where we have two discrete populations out of the mold, out of one single distribution, will collapse. What we see is that the hybrids are less well able to grow in the benthic zone than benthics or limnetics, and less well able to grow in the limnetic zone than the actual limnetic. So an individual with a trait in between the two ecotypes is less well adapted. So if you take that scenario and run it forward through multiple generations, you'll see, or you can say, that only the individuals here and individuals here are the most likely to survive. They're the individuals that are most likely to grow well, have large gonads, produce lots of babies, and over time the population will move towards these two discrete units. We can then run that on and say that, okay, so these individuals, the limnetic form, spend most of the time in the open water. The benthic form spend most of the time there in the shallow. The shallow water is warmer than the deep water. That means that those individuals are growing faster. They're they're generating their reproductive tissues, their organs are generating earlier in the year. Over time, the limnetic form will spawn later in the year than the benthic form. And then we start to get a temporal reproductive isolation between these two. Then we can start to get to a situation where, as well as individuals having a preferred niche or being adapted to their niche, they're now reproductively isolated from the other form. And we see that starting to happen in these sticklebacks. The hybrids between the two morphs are less well adapted. There's a reproductive isolation builds up over time between these two different morphs, between two different ecotypes. And at that point, we can start to move towards two distinct species. This is happening, This is happening in Europe, and some of the systems that I used to work on, where we've got this wonderful fish here, the European whitefish, and in some large, deep post-glacial lakes in Europe, we've got 4 distinct ecotypes in some of these lakes. Arctic char, which is another lake trout, another species which inhabits some of these lakes. We get the same trait, the same type of populations with multiple distinct ecotypes. These are lakes which were essentially became opened up at the end of the last glaciation. They're still extremely cold habitats. Not many species can live there. So we've got a benthic niche and a pelagic niche available for individuals within the population to evolve into. Once we get that, some of these individuals will then become piscivorous, they'll become predatory. So that's another niche that they can evolve into. And you get some really deep water individuals. And each of these populations, each of these subpopulations, has a distinct spawning time, feed or reproduce at a different time of year. And we get a point that these individuals, or this population, actually exists as subpopulations, as different ecotypes within or different forms of this whitefish. And these are characterized here by their gill rakers. Exactly the same thing that we saw in the sticklebacks. Some individuals, or one of these types of morphs, has many long gill rakers, adapted for pelagic feeding. Another type has very few, adapted for benthic feeding. They spawn at different times. If you stick them in a tank, set the conditions right, they can still hybridize. But in their natural conditions, in their natural environment, they don't. At what point do they become different species? Some work has been done with these complexes in the Alps in Europe, where we've got a combination of ecological and geographic isolation, where they're separated into distinct lakes, but they're also, so all of these are alpine lakes separated within a mountain range. A fish cannot get from here to there, cannot get from there to there. But they're evolving into these separate niches within the lakes, but they're also isolated in different lakes. So there's lots of potential for evolution here. What we're starting to see more recently as these lakes have become more productive through more people, more higher human densities, the lakes are getting warmer and more eutrophic, more productive. That means they're losing the benthic and pelagic components of the habitat. These lakes are entirely now fueled by pelagic production. Think of them as moving from a nice clear water lake to a big pea green soupy bowl. There's only one type of resource available, and what we see is that nice segregation in gill rakers is breaking down because there's no benefit to having fewer gill rakers or more gill rakers. So we're moving, like we did with the finches, through one individual or one population. It's the abundance of the frequency of different traits moving through time. Here we're seeing this in a species complex, that which one species had evolved into multiple different ecotypes. The environment has changed. That complex is breaking down through natural selection. This is the process through which selection is acting on genotypic variability. Okay, we'll pick this up again on Monday, and